Observe the plasma membrane under high magnification, and the idea of a solid, static boundary disappears. Beneath what looks like a fixed surface is a three-dimensional liquid, constantly undulating and rearranging its own molecular components. We know the cell is bound by this liquid because of a landmark study by L.D. Fry and Michael Edidin. They used a Sendai virus to trigger the fusion of a human cell and a mouse cell, merging two distinct biological identities into a single hybrid. Initially, human and mouse proteins remained strictly on their respective halves, but after 40 minutes at 37 degrees, they entirely intermixed, forming an integrated ring. This result provided the first direct visualization of lateral protein movement, showing proteins diffuse freely through a liquid environment. This confirmed the fluid mosaic mile. It describes the membrane as a shifting sea of lipids, embedded with a functional patchwork of proteins that move and interact within the bilayer. The foundation of this system is the phospholipid bilayer. Because the lipid tails are hydrophobic, they aggregate to hide from water, forcing the molecules to spontaneously self-assemble into a secure two-layered sheet with an oily core. This aversion between water and oil creates the membrane's basic structural logic, providing a self-healing barrier that automatically reseals itself if the cell surface is torn. To understand how a cell manages this liquid boundary, we have to look at the three forces at play: the physical movement of lipids, the chemical modifiers that control fluidity, and the embedded protein machinery. Because the lipids are held together by hydrophobic exclusion rather than rigid bonds, they are in constant motion. They exchange places laterally with their neighbors millions of times per second, a process that requires almost zero metabolic energy to maintain. Moving a lipid from one side of the bilayer to the other is a different story. This chart shows the energetic bottleneck of transverse diffusion. Dragging a water-loving head through the oily core requires a massive input of energy to strip away its hydration shell. To overcome this barrier, the cell employs the enzymatic translocators listed here. Flipases and flopases are ATP-dependent enzymes that manually move lipids across the core to maintain the precise asymmetry required for cell signaling. This movement must be precisely regulated because of environmental threats. If the temperature drops too low, the lipids pack tightly and the membrane freezes. If it gets too high, the structure becomes disordered and begins to leak. Organisms actively adapt by re-engineering their lipid tails. In cold environments, they produce unsaturated fatty acids with permanent kinks. These kinks physically prevent the lipids from packing into a rigid, crystalline state, as shown in the fluidity panel here. Animal cells utilize cholesterol as a primary regulator. As seen in this section of the diagram, the bulky cholesterol molecules wedge between the phospholipids. This restricts excessive movement at high heat to prevent leaks, while simultaneously preventing the tails from locking together in the cold. By constantly adjusting these molecular ratios, the cell ensures its membrane remains flexible enough to function without losing its structural integrity. While the lipids provide the fluid environment, the proteins provide the utility. These molecules make up the mosaic and are categorized by how deeply they are anchored into the bilayer. Integral proteins use hydrophobic alpha helices to span the oily core. Extracting them requires harsh detergents like SDS. Conversely, peripheral proteins do not enter the core, associating with the surface through weak ionic or hydrogen bonds. This surface-level attachment means they can be easily removed with mild lab treatments, such as changing the pH or increasing the salt concentration of the surrounding fluid. These swappable peripheral proteins and fixed integral channels transform the lipid bilayer from a simple barrier into a sophisticated array of biological sensors and gates. Beyond the standard model, evolution has modified this architecture to solve specific survival challenges or exploit the vulnerabilities of other organisms. For example, fungi utilize ergosterol instead of cholesterol. As shown in the center panel, modern antifungal drugs exploit this subtle molecular difference by binding specifically to ergosterol, physically tearing holes in the fungal membrane while leaving human cells intact. In mammalian red blood cells, the membrane requires even more reinforcement. Because these cells lack internal organelles for support, they rely on a two-dimensional internal protein network of spectrin and ankyrin to maintain their shape. This protein network dictates the cell's entire physical geometry. Even a single genetic mutation in the spectrin or ankyrin sequence can collapse this tension, resulting in fragile, spherical cells that characterize hereditary spherocytosis. When intact, this architecture allows the cell to aggressively deform and recover its shape, surviving the high-pressure transit through capillaries narrower than the cell itself. This dynamic architecture is the mechanism that allows a cell to maintain its integrity, resist infection, and move through the complex environments of a living body.